A few words about the matrix metalloproteinases or collagenases. Currently, there are 24 known MMPs identified in vertebrates, including 23 in humans. Their activity is low in normal conditions, but increased during repair or remodeling processes and is in diseased or inflamed tissues. MMPs are produced by multiple tissues and cells, and they're tissue specific. MMPs are secreted by connective tissue, pro-inflammatory, and other kind of cells, including fibroblasts, osteoblasts, endothelial cells, vascular smooth muscles, macrophages, neutrophils, lymphocytes, and so on and so forth. MMPs are regulated at multiple levels, including microRNA expression, activation of proenzyme to the active form, and the counteracting actions of endogenous tissue inhibitors of MMPs. And when matrix metalloproteinases are activated, they may lead to pathological process in different organs. For example, in liver or arterial walls, or as you may observe on this slide. But you shouldn't consider them only as the bad guys, okay? MMPs are involved, among other, in trophoblast implantation, embryogenesis, bone growth, angiogenesis, wound healing, and tissue regeneration. They can work kind of vice versa. So in the same type of tissue, MMPs can work as the good and the bad guys, and sometimes they work against each other. So it's a huge myriad of the working of MMPs only. And so every organ has an ACM with the unique composition that is generated in early embryonic stages. The function of the ACM goes beyond providing only physical support for tissue integrity and elasticity. It is a dynamic structure that is constantly remodeled to control tissue homeostasis. The functional importance to the ACM is illustrated by the wide range of tissue defects, or in severe cases, the embryonic lethality caused by mutations in genes that encode components of the ACM. Loss of function studies have also shown the importance of ECM proteins in developmental processes, as genetic deletion of specific ECM proteins such as fibronectin and collagens are often embryonic lethal. Okay, so now I'd like to show you, let's put this way, the main functions of the ACM. There are, of course, plenty of others that we're not going to, mostly not going to cover in today's lecture. But I would say this is the main ones that you see on the screen. So, scaffold function. Scaffolding is important for tissue renewal. Integrity of the ACM is critical for the organized regeneration of the tissues. Therefore, disruption of the extracellular matrix leads to impaired tissue regeneration and repair, such as, for example, cirrhosis of the liver, which is the consequence of persistent and progressive fibrosis, and we are going to touch on fibrosis in the following part of our in the following module. Next is the establishment of tissue microenvironments, because ECM controls the cell proliferation by acting as a depot of different growth factors, by binding and displaying growth factors and signaling through cellular receptors, hormones, and peptide production. And another important function is mechanical support, because mechanical support and mechanical transduction of the signal, so we call it mechanotransduction, because ACM is responsible for cell anchorage, cell migration, and maintenance of cell polarity. Cells also constantly check for and incorporate signals from ACM through mechanotransduction in the cytoskeleton. So it's also they also change their microenvironment by building and rearranging the ACM. So even the ACM also is involved in the clearing out of the cellular waste or debris, and it's also happened through mechanotransduction. Now, All this process, all these proteins provide the necessary support to cells and tissue, making, as you see, 3D structure of the ECM. Here you see the cell in the context of its environment. There are two main types of ECM: the interstitial connective tissue, as you see it over here, and the basement membrane. So as you see, the basement membrane is here on top of this, in top of interstitial connective tissue. All right, so interstitial matrix presents in the spaces between cells in the connective tissue and between the parenchymal epithelium and underlying vascular and smooth muscle structures. It is synthesized by mesenchymal cells, for example, fibroblast, and forms this 3D-dimensional amorphous gel structure. The interstitial matrix surrounds cells and is mainly composed of collagen number 1, then it's elastin as well, and then its third component, big component, is fibronectin, which provides structural scaffolding for tissues. By contrast, the basement membrane is a specialized form of ECM separating epithelium from the surrounding stroma and controlling cell organization and differentiation through interactions with cell surface receptors and ECM proteins. Synthesized by contribution of both underlying mesenchyme and overlying epithelium. The basement membrane has more compact than the interstitial matrix, but porous structure, something like chicken wire mesh, I would say, and mainly consists of collagen type 4, laminins, and some protoglycans and some other proteins that are synthesized and secreted by epithelial cells, endothelial cells, and underlying integrin expressing myofibroblasts. Basement membrane expresses different receptors, all right, such as integrins, for example, or that bind to ECM proteins. I'd like to emphasize one more time that every organ has its unique ECM composition, and the composition itself is a subject to change under different circumstances. Cells also continuously monitor and integrate the signals, right, that we already talked a little bit about it, from the ECM via mechanotransduction through the cytoskeleton.